Hello there. This movie will show you how to easily and conveniently grow Arabidopsis for analysis in the lab. This movie is designed to be used with the plant tracer system to screen for mutants in plant movement. Arabidopsis is the genetic model plant system. Arabidopsis was chosen because it has a small genome, it's easy to grow, makes a lot of seeds, and those seeds can be kept for a very long time. It also has other advantages, such as its small size and its well-sequenced and annotated genome. To cultivate Arabidopsis, only a few things are needed. These items include lights, soil, and in this case we're using these Jiffy 7 pots, a flat for holding the plants, and just a little bit of fertilizer, and in this case I use Miracle Grow. The Jiffy pots are soaked in water for about an hour. You'll notice that the pots are shaped as a flat disc. When placed in water, the discs will increase in volume approximately 5 times its original size. Once hydrated, the pots are placed into a flat at even spacing. Arabidopsis seeds are very tiny and will remain viable for many years when stored in wax paper envelopes to prevent sticking from electrostatic interactions, and you can keep them in the refrigerator. The seeds are kept in an airtight closed container with dryerite, that's calcium chloride, to absorb humidity in the refrigerator and maintain viability. If you're receiving Arabidopsis seeds from the ABRC, the Arabidopsis Biological Resource Center in Ohio, they will come in a plastic tube. The seed tubes from the ABRC have a stock number. You will receive a tube that has the wild type control, which is Columbia, and a variety of mutant lines. These mutants have mutations in specific genes that you will be testing to see if they have a functional role in Arabidopsis movement. The loss of the gene may give a phenotype that is different than the wild type, which may allow you to infer what those genes are doing. When sowing Arabidopsis seeds, gently tap the envelope into a folded square of wax paper, such as whey paper. Very gently sprinkle the seeds onto the wax paper. This way, it's possible to see and count how many seeds are being dispersed. When the desired number of seeds are on the wax paper, gently pick up the wax paper, bending it along the crease, and gently sprinkle it over wetted Jiffy 7 pots. Plant about 5 seeds per Jiffy pot. Make sure the flat is dated. Saran wrap or a clear dome will keep the Arabidopsis plants moist while the seeds imbibe or absorb water to begin the process of germination. The plants are grown under 24-hour light, such as fluorescent cool white works well with the Arabidopsis, or you can use specifically the lamps that we have here. After 3 days, tiny green Arabidopsis seeds can be visualized in the Jiffy 7 pots. They will continue to grow quickly and after 1 week should appear as small little plants. Arabidopsis is watered from the bottom, meaning that the water is never poured onto the plants, especially when the seedlings are very young. However, Arabidopsis plants do not like to sit in water as it's inhibitory to its growth. Plants' roots need air for respiration. So, grow the plants in a double flat system. An upper flat has a few small leaky holes in the bottom, and the lower flat is intact so that it acts as a reservoir. Soak the Jiffy 7 pots in the water for about 15 minutes. Then lift the upper leaky flat out of the water and place it into the new intact flat. This way the pots are moist but not saturated, enabling healthy root growth. Do not let the Jiffy 7 pots dry out or the plants will wilt and never fully recover. One sign that the Jiffy 7 pots are drying out is that the color of the pot goes from black to almost lighter brown. To avoid having the plants dry out, follow the watering regime. Monday and Wednesday, follow the dipping method. Friday, leave the plants with a soak in the water to ensure that they will not dry out. After 1 week, fertilize the young seedlings with Miracle Grow, which has micronutrients. Add just a pinch of the fertilizer powder to the water and then add this water to the flat. Arabidopsis grows as a rosette, which means that the leaves radiate out from a central point. After approximately 5 to 6 weeks, Arabidopsis will bolt, when a single central shoot will emerge bearing individual buds that will open up into flowers. This is the inflorescence stem, or the flowering shoot. It is this shooting structure that is of interest of the Plant Tracer project. This shoot is the organ of interest for movement studies and a target to screen for mutants in gravitropism and circumnutation. When the flowering shoot is approximately 4 to 6 centimeters tall, it is ready to screen for mutants that are impaired in movement. Find a wild-type control Columbia plant that is the same size flowering stem as the mutant. To screen for mutants in gravitropism, choose 2 Arabidopsis plants with young, growing, flowering stems that are about the same height. One plant genotype is the wild type and the other plant is the PGM, positive control. Or one plant genotype is the wild type and the other plant is the mutant of interest, which you're screening to see if there are changes in its movement properties. Place one of each of the plant genotypes resting on their sides with their foliar, that is the flowering stem tissue, facing towards each other. Make sure to have a dull black folder or dark velvet in the background. To set up the camera, open Lapse It. Press the capture button. Position your phone on the long side. Use clips or something heavy to brace your camera so that it is held fixed and the flowering stems are in the camera viewer. Be careful to position your plants so that they are in focus. You can tap to make sure they're in focus and that they are not too far away from the camera. Make sure that the genotype labels are visible in the camera viewer. To do this, I use a piece of labeled colored tape wrapped at the end of a paper clip. Make sure nothing disturbs the setup during the recording. It is easy to forget about the plants and bump into the recording device. Make sure that a metric black ruler with white writing is placed in the same focal plane of the inflorescence. Write down the names of your mutant and note if the apex is facing left or right. It's best if the names of the plant lines are visible in the camera viewer. Start the recording by hitting record. After 1.5 hours, you are done and stop the recording. Complete the movie with the stop button. Now export, publish, and save the movie to your photo roll, and it's ready for analysis with Plant Tracer.